y hemos hecho algunos tipos de contribuciones juntos en el campo del dolor. Hoy él nos va a ilustrar en su conferencia acerca de las últimas tecnologías que está aplicando para profundizar un poco más en este campo de la neurobiología sensorial. Y fundamentalmente tecnologías que implican el uso de técnicas de semi-high throughput, de alto rendimiento, para caracterizar la diversidad neuronal que existe en este campo. Para aquellos que no lo conocéis, Tim es un bioquímico fundamental que obtuvo la licenciatura en bioquímica por la Universidad de Berlín y luego estuvo realizando distintas estancias en la Universidad de Colonia, en el Weizmann Institute en Israel, en la Rockefeller University, en el Max Planck Institute of Neurobiology y en la Universidad de California en San Francisco, donde realizó su postdoctoral con John Lin en todo esto, en toda la temática de la señalización vía los nociceptores sensoriales de la piel. Actualmente él es profesor del Departamento de Anästhesiología y Terapia del Dolor en la Universidad de Colonia y donde está centrando sus estudios fundamentalmente en la aproximación esta de la microscopía semi-cuantitativa con el fin de estudiar los nociceptores desde un punto de vista un poco distinto al que nosotros hacemos, que es de forma individual, estudiarlos de forma de ver las poblaciones de esos receptores, cómo funcionan en respuesta a los distintos estímulos que se les está aplicando. Eso es importante porque cuando uno trabaja en nocicepción, lo primero que se da cuenta de que las neuronas no son idénticas, sino que tienen una distribución y una diversidad brutal, que eso complica el análisis y las respuestas. Y eso también es obviamente lo que suele ocurrir con los distintos umbrales de dolor que tienen los distintos individuos. Cada uno de nosotros tiene un umbral distinto porque está codificado de forma diferente por toda esa diversidad neuronal. Y por lo tanto es lo que llamamos una visión más de biología de sistemas que de individualidad de las neuronas. Y ya sin más, I just made a short introduction of your CV and what are you interested in your laboratory by using this semi-quantitative microscopy approach, and I just advanced to them that we are going to address all these topics. And I also asked Tim just to present the results in a very communicative way, because we have a lot of students, and instead of going to the very specialized, just to try to disseminate the topics that he is interested in, just at low level, just to understand, the students can understand all of what he's trying to address in his laboratory. Welcome to Alicante, welcome to Elche, welcome to Humboldt Institute, and we are just looking forward to this presentation.
Thanks a lot for that very kind introduction. It's really a great pleasure. I've actually been barely to Spain at all, so it's really very, very nice for me to be here. I learned from an older colleague of mine that you should try to give a talk so that afterwards everybody is asking himself, why am I not working on that? So, but after having seen quite a bit here, so please stick to what you're doing. But I'd like to introduce an aspect which might not be that familiar to you. Also, I think it is an aspect in pain research which is not really that much at the focus, namely signaling, intracelular signaling, which established sensitization. So that, as it's not that much in focus yet, please excuse this Edward somewhat bold statement that, of course, that is the most important part. What I'm going to show you is a little bit how kind of the general flow, I guess, of pain research, how we start from a general phenomenon, pain, build up on what other people have established, some mechanisms, then how to break that down on the cellular and in biochemical level to understand a little bit more details, and then actually go from there to understand novel mechanisms, go all the way back up to even humans, and thereby I hope to introduce a little bit the need for novel techniques, for novel aspects to be looked at, other than what is a common approach. I will introduce this high-contact screening microscopy technique and some new questions which arise from there. So I guess you Okay, I guess you heard more than enough introduction into pain, so I guess it's obvious what pain is, but there's also this kind of pain, and I'm especially, my group is especially interested in the second one, while here it's more about the input structures, what are really the sensors for the physical input. We believe that, especially for clinical pain, the major interest is in really how is that modulated, how are neurons sensitized, and why are suddenly physical stimuli which are otherwise not painful perceived as being painful. For example, high temperature, well, normal temperature during your bath, or actually more important, actually mechanical pressure, be it even just the pulsing of your arteries, which suddenly induce pain. So therefore, our quest is to understand really the mechanics behind that, and not so much into what are the structures which receive it, or the output structures in the brain. And how to study that? Well, the question is, what drives pain? Let's start really a little bit literally. So if you want to understand from an engineering side structures like that, the general approach in the pain field has been driven mostly actually by two techniques. One is behavioral science, the other is electrophysiology. Behavioral science gives you much insight into extracellular mediators, which modulate the behavior of these machines, while actually the electrophysiology is very much focusing on these round things, on these ion channels, indeed both aspects which very much explain behavior. Actually, I had a colleague of mine, very much a race enthusiast, who told me there's indeed a whole science behind what wheels to use when, and that it can very much explain you the transition from this basal state to this somewhat excited state there. So, but nevertheless, from an engineering side, I guess looking at these kind of structures, one might be tempted to also look at other things like, wow, is there an engine in this thing? Especially from here. And also, of course, can I shift gears somehow? And from a therapeutic aspect, of course, are there actually also brakes which are built in, which might be helpful, actually. It is my belief that actually much of these structures are encoded actually in the intracellular mechanisms rather than just purely on the ion channel world. So I'm a little bit too stupid to work on pain in the brain. I prefer this somewhat a little bit more simple structure of the peripheral sensory neuron. So of course that's where the subtle or not so subtle stimulus is perceived. And as I indicated, what we are interested in is why are suddenly easy, normal stimuli like this brush with a feather is perceived as painful. We are trying to go after that by looking at single neurons, at dissociated DRG neurons, and for us the picture is like that, so a large number, this is only a fraction of the extracellular components which are known, are described to actually result in sensitization. When I started working on pain some 10, 12 years ago, I was really puzzled because when you actually had a look at what kind of receptors these components activate, mostly that hasn't, at that time, had not been described, so it's not really so much the focus, but also the ones which are described or which you could get from other cellular systems. It's striking that basically any kind of input into these sensory neurons results in sensitization, which is somewhat mechanistically troubling. On the other hand, then of course electrophysiology has in the meantime established an ever-increasing number of ion channels, again this list is not complete, which are basically effector molecules of the sensitization process there. And well, it's not so much a white spot on my map here, but a yellow spot. The in-between is rather surprisingly ill-defined. So how are actually really these extracellular mediators result then in intracellular processes which in the end affect, among others, ion channels? These components here are basically the most common components of signaling pathways you could think of, so this does not really explain much. And on a more mechanistic level, actually, this huge variety of inputs and this huge variety of outputs, to me, question which we then pose to the field, but it's not really taken up yet, is, well, if you have so many inputs, is there then a discrete signaling? Are there parallel signaling pathways which are basically functioning on its own, resulting then basically in a very large number of separate, distinct states of sensitization? So is a sensitization induced by epinephrine something different than sensitization induced by TNF-alpha? Or is there actually convergence of these signaling intracellularly onto some modules, potentially one or more than one, so some overlapping mechanistics intracellularly, which would be, of course, pharmaceutically also interesting. You only need one drug to kill it all. Of course, financially, the other option would be nice, because then you need lots of drugs to treat all the patients. But it's a very fundamental question, and so far the literature cannot really even give an indication which of these two models is true. I started out in the laboratory of John Levine, and basically for that reason I was interested in one of these signaling components, because the epsilon, a protein kinase, a novel one, so it's not calcium dependent, and it's first of all shown to be involved in a large number of different pain phenotypes, sensitization phenotypes, which are associated with mechanical hypersensitivity. A system that here, what is nice about this protein kinase is that the process of activation goes together with a translocation to a membrane in sensory neurons. This membrane is actually the plasma membrane. The phenotype is not really as strong as depicted here. In average, it's much lower than that. Nevertheless, that gives you a handle to observe the process of activation of this signaling cascade in primary sensory neurons. And that was an assay which I developed in this laboratory, and which suited me nicely for a couple of years to investigate then the signaling pathways resulting in the activation of PKC epsilon. In his laboratory, much work has been done on epinephrine, being an activator, among others, of PKC epsilon, inducing PKC epsilon dependent sensitization, and I just tried to establish the signaling pathway in between. I sum this up here. It's some pharmacological work, and to my great surprise, that was actually really the very first real signaling pathway analyzed in a more meaningful manner. It also was very nice because it actually is the first time proved that these most classical signaling cascades, the CMP intracellular signaling cascade and the PKC system, can actually directly interact. Up to then, they have always been considered to be separate. And this is really nice. It also actually is the first proof that EPEC, a novel component, a CMP-dependent component, has a biological function. So lots of new stuff there. It's still highly cited, not exactly by the pain field, especially the brain. People like this kind of study, because in the pain field, it's always then the question, well, it's fine, you have something there, beautiful, but what is the ion channel that is regulated by that? And so in the end, I had to turn also to an ion channel to be recognized as a pain scientist, which I find a little bit funny. Nevertheless, one ion channel that everybody, I think, comes up with if he thinks of ion channels in the pain field is TRPV1. I guess I do not have to use it here. It's a serine 800, which has been shown by others to be phosphorylated by PKC epsilon. At that time, I got in contact with Shalan Goswami, then in the lab of another Hucho, actually. He's been the last PhD student of my father, actually, up to then working on something completely different than pain. That was something unexpected, that our interests would converge. Nevertheless, that suited me nice. Shalan found an interaction of tubulin with the C-terminus of TRPV1. And I was interested then, I recruited him into my lab as a postdoc, and I was interested to what extent actually these phosphorylation as S800 influences this interaction. Here, first of all, just a brief proof of principle. So if you express simply, easily the C-terminus of TRPV1 and pull that down, indeed you can pull down tubulin with it. It is a little bit tiny bit calcium-dependent process, but not very much. Shannon went then on to identify the interaction domains. He found actually two stretches which were most important to that. And if you have a look at the schematic presentation, this is indeed a very interesting region, as this is very, very close to the PKC-epsilon phosphorylation site. Together then with Gregorio here, with the help of him, or basically not help, but he actually produced this beautiful conformational model where indeed he found that tubulin can interact with the C-terminus. Interestingly, not the whole microtubules, but actually only the protofilaments, and there especially the beta-tubulin that fitted exactly to predictions from our biochemical work actually. So most likely TRPV1 sticks really to the very end of microtubules and is connected to that. As you can immediately see, this interaction model also is very interesting in terms of the phosphorylation side. You might have seen that little reddish ball in there. So that's serine 800. So right in the middle of this interaction, potential putative interaction domain there. And one might indeed therefore speculate that the phosphorylation could change that, this interaction. And that was indeed the case. If you now purify, I get a C-terminus of TRPV1, you in vitro first phosphorylate with PKC-epsilon, which was spiked a little bit with radioactive ATP. Then you see, okay, indeed, if PKC-epsilon is in there, you get a phosphorylation of TRPV1. And then you also see that thereby the interaction with tubulin is reduced. It is not too much, but it is very significant, so about 20-25% decrease. We hypothesize that actually this low number is due to the low efficiency of the in vitro phosphorylation of TRPV1 by PKC-epsilon, something which very often is the case. So therefore we did a precipitation of microtubules and then tested what kind of TRPV1 is interacting with that. And as you can see here, only non-phosphorylated TRPV1 can be pulled down with microtubules together. And that's a very, very sensitive assay. So we exposed actually the phosphoimager plate there for all the week, and no signal whatsoever came up there. What's the implication then on a cellular level? Shannon showed earlier in a different work that actually the interaction with TRPV1 stabilizes microtubules, actually to that extent that even leucodazole or high calcium does not really depolymerize microtubules anymore. So we therefore hypothesize that if you lose that interaction because you phosphorylate TRPV1 in the cell, that therefore you might see also a change of the microtubular structure in the cell. If you express TRPV1 in any various cells, it has very often a pretty funny effect on these cells. They have a much larger extent of philopodia, so somehow that is induced there, and you might see that here. So these little greenish extensions are actually TRPV1 labeled philopodia in F11 cells. And what is really striking is that indeed the microtubules go into all these philopodia. Usually they are formed rather by actin, and it's very uncommon to have microtubules in there, but apparently in the presence of TRPV1, that is not such an unlikely effect. And if you now activate PKC-epsilon, for example, the astrogen, which we showed to act directly on nociceptive neurons and activate PKC-epsilon in there, then indeed you have a tremendous reduction of these philopodia, fitting very nicely together with our hypothesis of TRPV1 stabilizing microtubules, and that the phosphorylation disrupting that interaction, therefore makes these microtubules more unstable, and therefore leading to a retraction of this phenotype. So, great surprise, though, the phenotype was actually much, much stronger. So if you have a cell here, again, that's F11 cells, so a cell line which was derived, immortalized DRG neuron, the cell line actually, which lacks completely endogenous expression of TRPV1, so pretty suitable for investigating the function of TRPV1. Here shown the tubulin. If you expose these cells to, again, an estrogen derivative activating PKC-absent, this has basically no effect on tubulin whatsoever. Nevertheless, as soon as you start expressing TRPV1 in these cells, again activating PKC-absent by G1, then you see a great loss of the microtubular network. So not only right at the membrane, but actually much, much even beyond that. And what was nice then, we've been of course testing, is it really PKCs which are involved? And yes, if you block PKCs, you do not see this destructive effect anymore. And even more so if you just introduce a single mutation, this serine 800, this phosphorylation site of PKC-absent. If you mutated that to an alanine, then again, an estrogen or G1 treatment of these cells does not change anything there. Well, some of you might know from the literature, PKC-absent is involved in the resensitization of TRPV1, changing the opening probability of TRPV1. So the instabilization of microtubules might be a very, very straightforward process. So you have more openings of among others, calcium channels. Calcium enters the cell, and microtubules are very sensitive to calcium, so that could be a very straightforward, easy explanation of what's happening. So we tested that, and this seemed not to be the case. Actually, the destruction of the microtubular network could also be observed when you were expressing not the full-length TRPV1, but actually only the C-terminus of that. And it could also be observed when you were buffering out all the calcium with butter AM, for example. And it could also be seen if you added ruthenium red to the mediums, or blocker of calcium channels in general. And ruthenium red by itself has not really much influence on the microtubular network. But if you have that there, and you again add estrogen, then again you lose everything. So indicating that the opening of the TRP channel is not really necessary, or is not really the cause for the destruction of the microtubule, which is, to my knowledge, really one of the first indications that there is functionality of TRPV1, which is very, very important beyond it being a protein which allows ions to flow through the membrane. So this is something else, completely independent of the functionality of TRPV1 as an ion channel. So does anything of that has anything to do with pain? I mean, all of that was biochemistry and cell biology, and there we attempted a very big jump into the unknown. TRPV1, of course, is not really that much known to be involved in mechanical sensitization. I told you PKC-absent has been mostly established as one of the mediators resulting in mechanical sensitization. And on top of that, not really much is known about microtubules in pain sensitization at all. One aspect which is known is that taxol, this microtubule stabilizing agent, actually results in neuropathic pain induction. John Levine did a couple of experiments for us. He injected antisense oligonucleotides into the spinal cord of rat, and thereby downregulated TRPV1 in DRG neurons. And as you can see here, it doesn't matter if TRPV1 is there or not there. You can always use with this compound G1, this estrogen compound, this PKC-absent inducer. You always can induce to a similar amount sensitization. If fitting nicely to the literature. Again, TRPV1 is not really crucial for mechanical sensitization. But if you now take Taxol and add that to that, as expected, it increases actually the sensitization. The great mismatch kind oligos, which only have a minimal effect, if at all, on the TRPV1 expression, reduces that effect a little bit. And now comes a big surprise: if you really take out major amounts of TRPV1 out of DRG neurons, then Taxol suddenly has the opposing effect. It's actually not only, it doesn't matter if Taxol is around or not, but it's actually blocking sensitization. And that's a very, very striking effect. I have to admit that there's quite a gap between our cell experiments and these behavioral experiments. Nevertheless, our data, which I showed you before, are very suggestive that we indeed might have an initial hint for the mechanisms which are underlying that. So what I showed you is that looking at signaling cascades can be extremely fruitful in finding novel mechanisms in very general terms, like the interaction between different signaling pathways, but it also can show you that actually the activity of compounds is very much context dependent. It depends absolutely not only on this one component which you're looking at, but always what kind of context, whatever that means in terms of expression, for example, of ion channels is around in that cell. So, what did I show you so far? Intracellular signaling in white spot on the map. TRPV1 interacts dynamically with microtubules. S800 phosphorylation of TRPV1 destabilizes microtubules. Downregulation of TRPV1 turns Taxol into an antihyperallergenic substance. So, ion currents, this is an ion current independent pain modulation. And in general, investigating signaling elucidates novel antihyperallergenic mechanisms. What I've also shown you on the side by that is actually that we now have more than one means to activate PKC epsilon. In the very first part I showed you epinephrine is doing it, but I now showed you that also estrogen is doing it. And actually, of course, from the literature you might know that also bradykinin, with a very different receptor system, is doing that as well, which indicates indeed that this one hypothesis which we put up, that there might be a convergence of various extracellular mediators onto a similar intracellular signaling machinery, which then established sensitization, might be true. And that was very exciting for us, and it opened also the opportunity to now ask a very different kind of question. Usually, if you think of what you're doing, and most likely, and most of the literature, they are always, one takes one component and tests them, and rightly so, because everything else very easily becomes rather difficult to work with. But in the end, this large number of extracellular mediators is there because they are there. I mean, if you have an inflammation, then it's an inflammatory soup. So there is more than one component in there. So therefore one has to question, well, how do these various sensitizing components play together? And so now having this convergence point, we started to do exactly that. So we asked, what happens to a sensitization signaling pathway if we basically stimulate it twice, if we do have two different inputs into that system? So to make that, bring that up to a little bit more schematic question is, what is 1 + 1 in terms of sensitization? Equals 2, or is the first sensitization already bringing the system up to a maximum? So the second one is just basically ignored, or what's going on? Again, we use this system which I introduced to you already, this little stretch of signaling cascade which we developed or defined. Again, this translocation of PKC epsilon to the plasma membrane is the major readout. But then we asked, okay, if we stimulate first here and then come in through a second route, can we activate PKC epsilon the second part? And this is one of the core experiments there. So we counted the number of neurons showing that translocation. It's the baseline around 5-6%. If you give a stimulus here, then it shoots up to about 22%. If you only give that stimulus alone, again, this shoots up to a pretty similar number of neurons. And now if you combine that, ta-da! Nothing happens. The quick answer is 1 + 1 = 0. And that is, first of all, very surprising. It gave me a very, very hard time to understand that. We've been working like crazy on that for 3 years, and it was one of these experimental series where always the control showed exactly the opposite of what you expected, and you always had to rethink the whole thing. It was nightmarish. On the other hand, though, it's an extremely appealing result, because that 1 + 1 = 0 is a very, very fast process. So if you only have 1 minute between these two stimuli, it is present already. But also if you have a time interval of 20 hours, which is in molecular cellular terms really very, very long, it stays there as well. So it's very unlikely that it's just one of these common internalization mechanisms of a receptor or something like that. And we took that in great length, and I will spare you all the details, to delineate that. What's the outcome of that is that any kind of first input into that, of course, activates PKC-epsilon, but on the side, of course, you have a branching here. Not only diacylglycerol is produced by phospholipase C, but also IP3, which in the end results, among others, in an increase of calcium and an activation of CAM kinase 2. And these are very, very interesting results. So we can now actually, by any kind of mechanisms of increase of intracellular calcium, or by activating CAM kinase 2, we can shift the cell into this non-responsive way. So thereby we can actually change and sensitizing extracellular mediator into something which at least gets ignored by the cell, and I'll show you a little bit later that it's not ignored by the cell, that it's actually doing the opposite. And that is very interesting, as actually Hogan et al. Showed that in neuropathic pain in DRG neurons, the calcium levels are reduced, which fits very nicely with here. Here we find any increased results in no pain. They found decreased results in more pain. And at that time when we figured that out, there was the first genetic model of CAM kinase 2 hyperactivity in the brain, which results in memory deletion. And there's actually only 2, maybe 3 mechanisms out there which show how memory can be lost. And I think we have to be the very first and the very first mechanistic hint in peripheral neurons how that could be accomplished. Again, the question is, all this is stupid cell culture, so does it matter for pain? And I only again show you the core result there, the core experiment. That's of course the behavioral experiment on the rat. So if you have first injection, you wait 30 minutes before you evaluate that, you see, okay, you get nice hyperalgesia. It makes you happy, it hurts, yes. And also, if you take a different kind of stimulus, the isoproternol, as I showed you before, also that alone results in nice hyperalgesia. Now comes again this combination of the two. It works that way. First you inject isoproternol, you wait 30 minutes, the hyperalgesia is fully established. Then you do the second incubation injection. Again, you wait 30 minutes, and then you measure your sensitivity levels, and it's down to 0. So it's not only that the second stimulus is ignored, but it's actually that suddenly the second stimulus is doing exactly the opposite what it should do. So therefore, the extracellular mediator, these things are not the bad guys. It's not the information in there that they are inherently sensitizing. But what makes them sensitizing is how the cell itself computes this input. And the cell has the ability to basically change the program, and by finding out a little bit about these mechanisms, now we are able to set the cell into a modus where inflammatory mediators, well, at least this one, is not sensitizing anymore, but turned into like the opposite, into an anti-inflammatory, an anti-hyperalgesic agent. which I find indeed rather stunning and also from the therapeutic side rather interesting. So we started first doing experiments on our own, and then we convinced a couple of students there that this might be worth pursuing. And it's basically the same kind of test like here, so you inject something, measure with a pressure algasiometer if there's pain or pain sensitization, and as you can see here, that's the case. And the second injection here in this example actually of exactly the same substance again brings it down to zero. And we now did that also in a different setting of sunburn on patients. There it works again, and we're currently extending that kind of approach to not only induce sensitization but also to patients who come to the clinic with painful incidences already. So again, summing that up, what I showed you is that this signaling pathway of course results in sensitization, but that also there exists endogenously in this cell a second branch which would result to desensitization, but this appears to be blocked in a native normal cell. But you can basically unblock that and flip the switch so that the very same input into the cell then results in that pathway and in desensitization of the cell and the animal. So what did I show you in the second part? Sensitization signaling branches. The pronociceptive stimuli can be inverted. The activation of a novel calcium kinase 2 dependent nociceptive module really deletes pain. And kind of on a conceptual level, the signaling state of the cell and the active signaling that rules about the output of the cell. So you can change the program, you can change how the output is calculated in a cell to put it a little bit more on an engineering level. So it's not a hardwired system in the cell, but you can actually greatly modulate the flow of information and therefore change how the cell interprets the input. This is nice, but of course creates novel trouble. Subgrouping of neurons usually is done either by its anatomy or by electrophysiologically by its firing patterns or responsiveness, or actually by its expression patterns of various ion channels in general. What our results now suggest is that on top of that, these cells can actually be in very different signaling states. And first of all, this is a qualitative thing. We now find signaling states are important. So next we would have to investigate can any kind of cell be in any of these states, or is it restricted to some of them? Can we reprogram all of these neurons, or I didn't delineate that, but we find this mechanism to affect basically only 25% of the cells. Is that a subgroup which is somehow special? So we have really to investigate that. So which makes our world now a little bit more complex. So it's not only anymore going by the usual candidates, looking what is expressed and then you know what's the function of that the neuron is. You really have to look out for more than that. Well, is that really necessary? And what's the trouble there? What is needed? Let's just go take a step back. What are we really investigating there? So if you have a look at a single cell, the possibilities of this cell are tremendous already. So we have about 15,000 genes which are expressed in a cell. So out of the 25,000 which could be expressed. So each of these genes, in average, comes with about 15 isoforms. We have more than 200 possibilities of post-translational modifications. And then comes combinatorics. So we have protein-protein complexes, so they are not alone. We have combinatoric of subcellular localization and combinatoric of relative expression levels. Did somebody sum that up? I don't know, I only came to the first 3, that's about 50 million, and so I kind of estimated that to be a lot. Okay, so it's a tremendous amount of different states, because each single thing would be a different state than of a cell. It could be the cause for flipping the switch from what is sensitizing a substance or desensitizing a substance. And now I'm saying, well, on top of that, don't forget about the history of the cell. Really look at what signaling has been occurred already. I mean, that's crazy. It's extending this multidimensionality again by a tremendous amount of possibilities. So is that really helpful? And actually, does that result in new insight? And for getting a grip on that, I'd like to consider you the following problem. Let's say you're a spy and you want to investigate how a foreign capital like Moscow is working. So there are quite a large number of satellites there, so you have pretty good insight there. Engineers build ever better satellites, so you get ever better resolution, so you see exactly where every house is, you see even where trees are, where cars are parking. Consider, I don't know, a stat-like improvement on the satellite level. You might read the books which are lying around there, whatever. So you can zoom in always better and better and define this whole city on a static level. And now I'm proposing, well, static is fine. The complexity of such a system would be enormous already. There's something like 12 million people living there legally and another 4 million illegally. And so then finding out the relationship of them, and it's tremendously complex. Could actually, looking at a time dimension, reduce the complexity of that. And I guess most of you have used Google Map already. All this is done with Google Map. And if you go to this traffic option there, that's exactly what I'm proposing. You look at the time dimension, forget about anything that is static in there. And that reduces the complexity of the problem tremendously. Because in the end, you don't care if there's a building or not, as long as it doesn't move. You know, and if it's a building and nobody's going there, why should I be interested in that? So it's completely, it doesn't tell you anything. So by looking at kinetics of processes, you reduce the complexity of your problem dramatically. You basically get rid of 99% of that complexity. And if you go, so in that case, you immediately get a map of where streets are and where used streets are. And if you go from a qualitative to a quantitative measure, by not just looking, is there movement, yes or no, but if you look, is there fast movement or slow movement, you immediately get where all the traffic jams are, and that's a very good indicator of where the centers of power are, where most people want to get to, and so you really get also mechanistically quite a hint what to focus on. And so a very similar thing we are proposing for signaling pathways and for the mechanisms of cells. And in terms of signaling pathways, that really means we would have to investigate them in all these different DRG neurons. I mean, basically nobody has done that so far, so we cannot use a model system, so we have to do population studies over all neurons, and that's a pretty difficult task. So that's why we actually started to with an automatized microscopy, and so since a couple of years now we have a high content screening microscope. Everybody of course sees immediately, yeah sure, this is a microscope. But if you open that up, it's actually really a microscope. They just chopped off the oculars and put it into this bigger box there, and that's it. The nice thing about our setup is that we have a liquid handling module on top of that. It has an incubation chamber which is fully controlled in there, so you can even do live cell imaging. And the nice thing is you can always go back to the very same cells after hours, after days and whatever, and you find them. And therefore you can really do multiple measurements, even fix them after doing calcium imaging, for example, fix them and look for expression levels of whatever proteins, for example. A very nice machine. So the general experimental setup is you kill a rat, you spread the DRG neurons on cell culture, you can then do object identification, quantify that, transform that into mathematical functions, and as all of that is really quantitative, and now we have a 14-bit camera, so that's 62,000 grade levels. It really is very, very informative and sensitive. We use that to monitor a couple of different components, and that the possibilities are pretty large. You can of course, anything that can be detected by an antibody, basically, or any dye reaction can be observed by that. The principal mechanisms, of course, again, is object identification. So this is one of these view fields, one of the examples of our culture. At least this is now one of the opportunities where DRG neurons are really nice because they are round, and round is very easy to define in mathematical terms. So very easily you can select the neurons from other things there. But what is obvious, of course, is that indeed you have a very, very big variability in these kind of systems, so from very small to very big, and from very bright to not so bright. And there it's really interplay that you have to do some quantitative approach. Here we combine that actually with using a phosphoantibody, just this MAP kinase ERG. So therefore we can actually follow quantitatively the activation pattern of neurons. Of course, antibodies, that's an endpoint analysis. So first you fix the cell and then you stain, so it's not continuous measurements, but over the population you get all these different levels. What you might appreciate already here is that variability of these neurons is really immense. I mean, you have a standard deviation of 200 to 300%. So that's pretty huge. I mean, with just counting plus and positive and negative neurons, so what usually kind of students have to do for months and months sitting there in the darkroom. That sensitivity is pretty low. On top of that, we as a group leader, we don't really know what you counted there. So, but with this machine, one can go back to the same data over and over again. And as you see here, if you then expose these cells, for example, to NGF, you see that the mean intensity, so the activation level, the amount of phosphorylated ERG increases dramatically. You can do that even in 2D, so comparing the expression levels of ERG with the amount of phosphorylation there. All this is done on a single cell level, and this increases again the sensitivity, because you see this is kind of an 8-fold increase from here. But if you would not have this dimension, then this would be still in the baseline of your results. So what is also nice is that now you can do that with large numbers of neurons. So we have a study which we currently try to publish, which went through over 1 million of neurons. And I don't have that many students. But it also, on top of that, of having more happy students in the lab, because they don't sit so much in the dark room, it's really quantitative. You can do power analysis. I mean, a general thing is you have your data, you send them in, and the reviewer says, well, beautiful data, but increase that your N, you know, more neurons to be measured. And, well, why more? I mean, I don't want that. Here you can really make a real argument. So we usually measure something like 50 to 100,000 neurons, and then thereby we get a very nice indication of how the population looks like. And then we can estimate how many cells do we have to count to depict that population in a decently nice manner. And so if you want to measure just one component, for example, this ERG component, then you need about 500 cells to have only something like 5% of error, which is decent. But it also indicates actually this approach then, what most of the people then forget, well, if you have these cells, well, throw a second marker on that, and then we get much more information. But that also means that you need much more cells. So if we add actually a second marker to that, you need at least 2,000 cells to 5,000 cells to reach the same level of sensitivity. And that's something mostly forgotten actually in most of the publications that we've seen, that it's not that simple to just throw a second marker on that. And it's really nice to fight back reviewers. You have now a mathematical proof of the sensitivity of your method. So what do we do with that? It's now opens you the opportunity to really do very defined, very precise measurements, for example, of those response curves here, PGE2 and PGI2. This is actually a pKa measurement which we have here. Or actually time kinetics, and these are really the very first kinetics, I believe, of endogenously expressed signaling components in primary sensory neurons. So, of course, we jumped onto a couple of known components. NGF would go up here, but then GDNF, isoproterenol estrogen, I introduced them to you. All of them are sensitizing. We just had a look at ERK activities, and we found very, very different kind of kinetics. What is nice, we had a study comparing that then with EGF. EGF shows some kind of activation, but not really that much. And indeed, EGF does not sensitize these neurons, while all these sensitize them. And this now poses a new question, actually, where in such a kinetic is sensitization coded in? I mean, there are many different possibilities how that could be coded. One could be really kind of a slope here, one could be an absolute value. It has to reach here this level, and then ping, the thing is sensitized. Or it could be kind of the area of the curve. I mean, you have much more experience with evaluating the quantitative data than I do. This kind of suggested that at least these later time points, 30 minutes and beyond, might be indicative of what is sensitizing or not. And we now took that and asked the question, can we now actually inverse the whole process actually of most of the pain research which is done so far? Mostly so far people start with a component, they inject it into an animal or throw that onto the neurons. They find some known sensitization activity like increased coherence or pain behavior, and then if they don't want to stop working, then at the very end they try to figure out why that's so and try to figure out an intracellular signaling cascade, for example. We now tried, can we actually turn that around? Can we start with intracellular signaling and then do all the one afterwards? And so we asked, okay, which components are known now in this is GDF, MGF, EGF, and then there are, in wounds there are much more growth factors than that. There's, for example, FGF, but also PDGF, IGF, and EGF, which is secreted by the skin in the course of the lesion. And so we did a kind of mini screen, if you want to use that word screen there, with the 5 components, but 4 components. But what it clearly shows you is that, well, yes, indeed, there are some components which result in a very strong activation of phosphor-ERK, and we then, this is all unpublished data now, we went the path and the other way around. We look, is the respective receptor expressed in DRG neurons? It is. It's actually in all DRG neurons, not only nociceptive ones. If we give FGF, this results in phosphorylation, and that can be blocked by an inhibitor of the receptor, so it's indeed a specific process. It also has this kind of kinetic which we found for other growth factors which are sensitizing. And it has a dose response curve again, just with one plateau and one turning point, indicating indeed one receptor process there. And then we asked the Waxman lab in Yale if they could do some recordings, and they found a very strong increase in the coherent density of the NAD 1.8. Again, a process which could be blocked to a great extent by an inhibitor of the MAP kinase. So our readout was the MAP kinase ERK. This is an inhibitor blocking most of this increase in density. And then eventually in the end, doing the behavioral experiment again, BFGF is sensitizing, strongly sensitizing, again a phenomenon which which mostly can be blocked by the ERK inhibitor. So, to sum this up, what did I show you? In the first part I showed you a typical way how to stumble into novel problems. I came from a completely different background and thought, well, pain is fun, and what do you learn during a PhD is about pain anyway, so why not continuing with that? And so I just simply thought about, well, do what everybody is doing in cells, looking for signaling cascades. Surprisingly, it's a basically forgotten aspect in nociceptive pain, so it's just upcoming. It gives you lots of indication. We filled up a little bit the space in here by indicating actually also other cellular phenomena which are involved in sensitization, like indeed the stabilization of microtubules. We could identify this interaction with SCRIP1 and the dynamic interaction and the consequences of that. Something I did not really allude to much is that this is a subgroup-specific phenomenon. It's not occurring in all cellular neurons, not in all nociceptive neurons, but only in these IP4 positive ones. I showed you that we could, by looking at intracellular signaling cascades, we can identify novel components to act on sensory neurons. So this was the first example showing that really oestrogen is acting directly on the sensory neuron. I showed you this concept that there might be something like a conversion of sensitizing signaling pathway onto a common sensitization machinery, which of course would be extremely interesting if one could further detail what belongs to that and what the mechanisms of that are. But I could also show that in addition to sensitization signaling, these substances also activate, to put it a little bit simple, but it's not really hitting the point, negative feedback loops. It's more than that. It's really the components, aspects of signaling which alter the information flow in these cells completely to that extent that a sensitizing substance turns into a desensitizing substance. And I showed you a little bit how we did that. So starting among others from biochemical data, going through cellular data, up to the human. I introduced you this still novel technique in the field, high content microscopy, as one of the solutions, how to fill basically the gap between behavioral pain research and electrophysiology with a meaningful cell biology. And I indicated that thereby in the end that now we can basically turn this whole process of identification, what components are sensitizing and what components might be desensitizing around, by starting now intracellularly, but looking at intracellular signaling cascade and thereby identifying all the components like FGF, what I showed you here. And of course, I'm not doing the work anymore. So I just moved from Berlin to Cologne, so this is a mixture of both labs. The work which I showed you was made basically by Christina Andres. She set up all these automatic microscopy approach. Recently is a current postdoc in my lab. He was working among others on PKA. Vanessa Zuko was a technician, which instead of a student I put into the dark room and I still looked at PKC epsilon. I got tremendous help by my former mentor, John Levine, who did all the behavioral experiment together with his postdoc colleague Gardina. I showed you some experiments which the Steve Maxman lab did for us in terms of energy 1.8 electrophysiology. And we got much help by Jan Hasenauer, and also in that context, Stefan Walter, to systems biologists with whom we collaborate very closely. So many thanks.
Thank you. Now questions for the students. Any curiosity, students? They don't know.
How long do you have to wait from first shot or whatever you did it, how long do you have to wait so the second one will have the same reaction, will have the same effects?
Well, it's like all behavioral experiments. It's not a kind of experiment where you can really do easily tons of different paradigms. So we basically only parallel to what we've done, or what usually has been done in rat behavior, and that's this half-an-hour time point. But from the personal experience, so it starts hurting immediately. It's a little bit difficult, therefore, to blindfold actually the students. I've all different kind of setups built there, but and our cellular data would suggest that it's basically immediately would change the response there. We only tested this after half an hour.
Because you were talking about time.
Right.
And then you show that graph that on different factors and the kinetics, right? So have you tried the opposite, the kinetics of going down somehow of those factors in the cell?
Well, these kinetics which I showed you were of ERG. So with ERG, currently we cannot really see this flipping of the switch. So that we only saw with PKC epsilon so far. And there, indeed, I mean, very, very quickly, again, after just 1 minute, we had this block of a second activation, and even beyond that, there's a little experimental window of opportunity to have the first stimulus being on the way of activating PKC epsilon, but it's not quite there yet. And then you add the second one, and you then read out if there's translocation at the time point when there should be translocation. It's a little bit difficult to explain. But there you see that indeed already then the second stimulus actively blocks the ongoing first pathway towards PKC epsilon. So there is signaling going on. It's not just receptor internalization or some negative feedback which cuts off the signaling input into the cell, but it's still the second signal starts the signaling. There's an active signaling which abrogates even the very first pathway down to PKC epsilon. And I did not really have much time to elucidate that, but you can actually now use the pharmacology which we established there to use substances which are not painful to flip the switch, so to result in an increase of intracellular calcium and an activation of the kinase 2. For example, like guanidine, it is not painful at all. Nevertheless, if you then come in and try to sensitize that via PKC epsilon way, it doesn't work. And even beyond that, you can even use these non-painful substances to even revert already established pain into the animals. So to sensitize them and then come in with a substance like guanidine that again deletes the pain in these animals. Any question?
Any additional question? Yes. Thinking about the application of your research into the development of new painkillers, is there any molecular target that you would pick or way too complex to this? Pick one more.
Well, I think that's pretty nice. It suggests a very general mechanism. I mean, it's really about the basal level of calcium in the cell. Calcium is regulated by a large number of components. Feel free to pick one. No, indeed. I mean, in the end, this could be also an alternative explanation of capsaicin patches, why they work. You know, usually it's just always this argument of desensitization of TRPV1, and therefore, as TRPV1 is causal for everything in the pain field, therefore there's no more pain, which I think is a little bit too straightforward of an explanation. But that could be an alternative explanation of that. So you have an ongoing, even in the desensitized state, nevertheless, your receptor is always exposed to capsaicins, so you might not get the full response anymore, and desensitization basically cuts off most of that the calcium influx into the cell. What would be interesting though is to have a look at basal calcium, if that is a little bit increased by having a continuous presence of capsaicin around. I actually think I've seen data to indicate that. And that would indicate, along with our mechanisms, that this would activate these CAP kinase 2 process. What CAP kinase 2 is exactly doing in there, I don't know. But therefore then it shifts the whole cell into making it prone to rather activate desensitization pathways than sensitization.
Any other additional question? Anna?
So, I don't know if you said so maybe you mentioned, but in your screening experiments with the microscope screening, so for you this discriminate between the neurons or another
but I did not indicate that. So we have multiple aspects there. So number one is what I indicated, they are nice, round, and usually rather big in contrast to the glia cells. So size is one of the aspects how we can discriminate between glia and neurons. The second one, you might have seen that somewhere on the slides, even though I did not point that out, use PGP-0.5 as a positive neuronal marker. So having also on one channel then an active fluorescent signal. Again, there you have to set thresholds, what kind of intensity you still consider to be a signal and what not. But that's another important parameter to identify those.
And then for the cells that are healthy or not healthy, in cells that are dead, for example, in our experiments in calcium, at the end you always use the potassium chloride to check that the cells are really live. So you also use it.
But if you do live cell calcium imaging, yes, in the end we're always doing potassium chloride pulse to see if they're active or not. Still alive or not. Yeah. For most of the analysis though is an endpoint analysis. So as I indicated, so we treat the cells for certain time differences, then we fix them, then we stain them. So therefore there is no real differentiation between live and dead.
Do you expect that volumetric channels are other than TB1 are interacting with microtubulars? TB1 channels? Because
yeah, there are also other channels interacting with microtubules. So we showed that also for TRPV4.
Before?
Right. We did not go beyond that, but otherwise, actually, there are also potassium channels interconnected to microtubules. I think it's a track channel. I think Sarah has shown that. So.
Not about you don't know anything about TB1.
Well, we've shown that interaction. Yes.
TB1.
TRPV1. Yes. Absolutely.
Because TB1 is also reported to be involved in mechanical hypnotism.
Yeah, there are not. There are not really that many reports pointing really into that direction. I mean, it's very clear for thermal sensitivity. The mechano aspect of that is rather weak actually. So I believe in that. I believe that TRPV1 has an influence also on mechanical sensitization. But I mean, the literature is not really that strongly suggesting that. Of course, I mean, the literature in the meantime is tremendously large, and you can find anything that you want in there. But I believe in that, but it's not the main, of course, the main target if you think of mechanical sensitization. It's rather TRPV4 that, and that's why we're looking into it.
Any more questions? Sakti?
I have a question regarding high context screening microscopy. You said you measure the neurons, many number of neurons. But could we differentiate, like based on the size, which which fibers have increased in expression of, like, for example, when you use this NGF. I mean, like A delta C fibers. So we have difference in the expression.
But of course, we cannot do subgrouping. The funny thing, though, is that there is not really a very clear-cut one-to-one translation from this anatomical differentiation in A delta, A beta, C fibers down to molecular markers. I mean, there are some markers which are usually used, like in TRIP1, CGFP expression, and stuff like that, but it's not an all-or-none marker. They are expressed in nociceptive neurons, yes, but they are also expressed in non-nociceptive neurons. And we just have gone through a pretty heavy battle with that. I mean, everybody is using them as markers for different classes of nociceptive neurons. And we were crucified for doing the same. Of course, there are some few percent which are also expressed in non-nociceptive neurons, so therefore cannot really use them as real correlates of A beta, C fibers, and then whatever. I mean, within each technique, you have very clear-cut differentiation criteria, anatomically, thickness, and whatever, electrophysiologically, on fiber speed and stuff like that, or on different expression of different ion channels and NVs and stuff like that, or of course even still connected to the skin, different responsiveness to thermal versus mechanical or whatever. But that does not really translate one-to-one then to the third kind of description, like protein expression. It's more, you need more than one component. So that's what usually is done, and that's still what's common in the field, and it's very valuable and it's very helpful. But one has to be aware you cannot just equal an anatomical differentiation with an electrophysiological differentiation with an expression profiling or expression marker differentiation of subgroups. But of course, with that, I mean, we go for expressions that we stand for TRIP1, we stand for CGFP, we stand for NAV of the hate, and we include these kind of common markers. One thing which I'd like to point out with this high-power screen microscope is actually, so currently we use a rather stupid, dull kind of analysis. It's really identifying the whole cell as a whole cell and integrating all the whole cell. Of course, you can go further than that. One can differentiate subcellular localization in these cells as well. Currently, we have enough trouble with the full cell data, so we're not really doing that. But it can be used, and actually for activation of transcription factors, for example, we're currently already also doing that. So you have translocation, for example, from the cytoplasm to the nucleus, that's very easy differentiable and read also from activation. So it is a very nice, suitable instrument, and I believe it can be really help tremendously to deal with these proteomic lists which we more and more get. In the end, we always have to opt to bet on one of these components to do real cell biology and electrophysiology with that, because it's so time-consuming to work with that. I think that can be a nice intermediate step to weed out which components to look at, which are really active on the neuron. Usually the way is the other way around. It looks is a receptor for that respective component expressed on the neuron, which is rather a difficult thing to do. Antibodies against receptors are inherently bad, actually, mostly, and receptors are rather low expressed, so it's very, very difficult to detect them. And even if it's expressed, it might not be active, it might be internalized, or it might be desensitized, or whatever. And therefore, we propose actually to go the other way around. Let's throw something on that and see, does the cell react? If it's reacting, there must be an input channel. And then let's see what is the receptor, or what's the way that this compound is using to activate the sensitization pathway. possibilities of detecting novel mechanisms quite dramatically. It's not always has to be then an ion channel, a cation channel, a trip channel.
No more questions? Thank you very much, Dean.
Thank you. It was
Very good.